Assalamualaikum. Let's continue with part 7 or the final part of chapter 10 lecture, Organization of Primary Tissues in Young Roots. As shown in figure A, the transverse section of a typical root. In the roots of typical gymnosperms and eudicots as well as some monocots, the stele is a vascular cylinder consisting of a lobe core of xylem with phloem between the lobes. So here it shows the magnified photo. The outside layer is the endodermis, followed by the pericycle. And then the blue stain rounded section here is the phloem, and then the purple stain structure here is the xylem. The phloem transport the nutrient, and the xylem mainly transport the water. And in figure B, transverse section of a root with parenchyma in the center. So the stele of many monocot roots is a vascular cylinder with a core of parenchyma surrounded by a ring of alternating xylem and phloem. So here the outside layer is the epidermis, followed by the cortex, and then a vascular cylinder, endodermis, pericycle, and then the core of the parenchymal cell in the middle, and xylem and phloem. So lateral roots arise from within the pericycle, the outermost cell layer in the vascular cylinder. So as we can see here, this is the cortex and the vascular cylinder, and you can see the structure here is the imagined lateral root. And the circular structure here is the pericycle. So from here, it will emerge, becoming the lateral root. For primary growth of shoots, a shoot apical meristem is a dome-shaped mass of dividing cells at the tip of the terminal bud. So these give rise to the repetition of internodes and leaf-bearing nodes. Okay, here we can see in the figure, this is the dome-shaped mass, apical meristem. And here are the leaf primordia, and you can see the developing vascular strand here, as well as the axillary bud meristems. For tissue organization of stems, in most eudicots and gymnosperm, which is the naked seed plant, the vascular tissue consists of vascular bundles arranged in a ring. So as you can see in figure A, a eudicot stem, for example here sunflower, with vascular bundle forming a ring. Ground tissue toward the inside is called pith, and ground tissue toward the outside is called cortex. So here the outermost layer is the epidermis, and then followed by the cortex. The ring structure here is the vascular bundles. Purple stain is parenchyma, which is the fiber cell. Then the blue stain here make up the phloem, and down here is xylem. So this one whole structure is called vascular bundle, arranged in the ring arrangement. So in the center is the pith. Different in most monocots. Okay, for monocot stem, the vascular bundles are scattered, no more arranged in a ring. It is scattered throughout the ground tissue rather than forming a ring. So here we can see in figure B, a monocot stem. For example, maize, with a vascular bundle scattered throughout the ground tissue. In such an arrangement, ground tissue is not partitioned into pith and cortex. So here, in this figure, it shows the epidermis on the outermost layer, and then the light stain structure are the ground tissue, and then the rounded structure are the vascular bundles, consist of the xylem and phloem, and it is scattered throughout the ground tissue. For tissue organization of leaves, so the epidermal barrier in leaf is interrupted by stomata. So stomata is like pore on the leaf, which allow carbon dioxide exchange between the surrounding air and the photosynthetic cells within a leaf. So you can see here, this is the magnified photo of a leaf epidermal. And we can see the stomata or the pore on the surface. So the ground tissue in a leaf is sandwiched between the upper and lower epidermis, and the vascular tissue of each leaf is continuous to the vascular tissue of the stem of the plant. Okay, here we can see in the figure the structure of the leaf. So the outside layer is called cuticle, where the stomata can be found, the stoma. And then this is the sclerenchyma fibers. And then we can see here the long cell tissue called palisade mesophyll. It is tightly packed. And then the bottom one is the spongy mesophyll. So this is more aerated tissue, contain air spaces in between the cells. And then the lower part is the lower epidermis. So you can see the gut cells that control the opening of the pore. And then here is the vein of the leaf. So this brown color are the bundle sheath cell. And then inside we have the xylem and also phloem of the leaf. So this is the surface view. Figure B shows the surface view of the spiderwort Transdensantia leaf. So you can see the stomata pore and the gut cells that control the opening of the pore. For secondary growth, so secondary growth add girth to the stem and roots in woody plant. The secondary growth occurs in stem and root of woody plant but rarely in the leaf. So these are mostly found in the stem and roots. So the secondary plant body consists of the tissue produced by vascular cambium and cork cambium. Okay, you can see in the figure, the outer layer is the cork, followed by cork cambium, and then the phloem. This made up the periderm. And then the secondary phloem, vascular cambium, as well as the secondary xylem. So the primary and secondary growth of a stem. This figure shows the growth in a 2-year-old stem. So the top is the new growth and the bottom is the old growth. So from the outside is the epidermis layer, followed by cortex, and then the primary phloem, vascular cambium, primary xylem, and the pith in the middle. So for the cross section, you can see here. So the old part of the stem now has periderm, mainly cork cambia and cork, and also secondary phloem, and also secondary xylem. So here we can see the layers of periderm, and then the bark, cork, and most recent cork cambium. So the growth you can see throughout the cross section. So this is another figure showing the primary and secondary growth of a stem. So this part is growth from 2 years ago, and then this is more recent, which is a year ago, last year's growth, and then this is the most recent, which is this year's growth of the plant. So that's why The stem at the bottom of the plant is more thick in comparison to the top of a plant. So we can see different layers for the secondary growth, the second year, the cork cambium, secondary phloem, vascular cambium, secondary xylem, which is second ring, and secondary phloem and secondary xylem. So here we can see the growth, secondary growth of the first year, and then the top is the primary growth, which is the current year. The vascular cambium and secondary vascular tissue. So the vascular cambium is a cylinder of meristematic cells. It is one cell thick and developed from parenchyma cells. So we can see in the figure, it shows the outer bark, and then the inner bark consists of the secondary phloem and cork cambium. And then we can see here the vascular cambium, the sapwood and the heartwood, and in the middle we can see the pith. And then the lines here show the ray of the growth, and the circular structure we can see the growth ring. So as a tree or woody shrub ages, the older layers of secondary xylem, and which is the heartwood, no longer transport water and minerals. So this part is already hardened, so it does not function to transport water and minerals. So the outer layers known as sapwood. So this region still transport material through the xylem. Okay, this is the sapwood, this is the heartwood. So here it shows another figure showing the structure of secondary plant body. The growth ring can be seen here, and the vascular ray, the secondary xylem made up of heartwood and sapwood, vascular cambium, and then the bark, which made up of secondary phloem and layers of periderm. Here's another figure showing the cross section of a 3-year-old tilliard stem. Similar structure. Secondary xylem, late wood and early wood, vascular cambium, secondary phloem, cork cambium, and cork, and the bark. So cork cambia and the production of periderm. So cork cambium give rise to the secondary plant body's protective covering, or the periderm. So the periderm consists of the cork cambium plus the layers of cork cells it produces. And then finally, the bark. The bark consists of all the tissues external to the vascular cambium, including secondary phloem and periderm. So this is the end of chapter 10 lecture, and these are the references, and thank you for listening.